Hello, everyone. Welcome to a extra special session of a monthly mastermind meeting. I have some wonderful experts today. If you're in the compounding world at all, you know that there are some proposed updates to USP 795 and 797 chapters. And so when these proposed guidelines came out, if you're anything like me, you probably didn't have time to fully read them all, or you read them all and you're like, okay, but where's the changes? What do I need to pay attention to? So instead of spending hours and probably misunderstanding what was proposed, doing it myself, I called up one of my favorite compounding consultants, Two Trees Consulting, to help digest all of these proposed guidelines and really narrow it down for me and you to help us to understand what are going to be the most impactful changes to USP 795 and 797. If you are not a compounder, this session is probably not for you, but you probably know a compounder and feel free to share it with them. We have Dr. Amy Summers with us, who is a fantastic compounding pharmacist that knows just about everything when it comes to compounding. And I'll let her give herself a formal introduction, but she is going to take us through and really digest this all for us. Let us know what are the proposed changes, which ones are good, which ones maybe aren't so good, and then how you can comment and how you can maybe affect some change, because these are just proposed. These are not final at this point. So I'd like to welcome Dr. Amy Summers in. Amy, please give everybody a quick review of where you are and what your background is, and then we'll dive into the content.
Sure. Thank you, Lisa. I am Amy Summers. I'm a pharmacist, graduate of San Francisco back in the days, and I was one of the first to get that BCSCP credential. I am involved pretty much straight up in compounding from the 503A side to even the 503B side. I've been a managing director of a 503B. I've been a pharmacist in charge at a handful of 503As. I'm a consultant full-time now, and working with Two Trees Consulting is a great place to start if you have any, questions or you're struggling with something. This is a good place to start.
Absolutely. Couldn't agree more. I know a lot of pharmacies are concerned about accreditation, but I don't think accreditation necessarily is for everyone. I think it's a great idea, but just being compliant with your state rules and laws and understanding, the guidelines, I think is really critical. And then Two Trees Consulting can help you in setting up a lab, expanding a lab, expanding services, if you've been doing non-sterile and maybe you see an opportunity in sterile, all those kinds of things. So please feel free to reach out over to May, who's my main contact over at Two Trees, but she has wonderful consultants in all different kinds of little niches, because, compounding niches down even further, like you said, 503A, 503B, sterile, non-sterile, all those kinds of things. So Amy, I kind of gave you a big task of digesting these updates and kind of dumbing it down, if you will, for us that really don't live and breathe the guidelines per se. We might live and breathe the day-to-day workings in a compounding lab, but take us through on what this looks like. I'm going to be monitoring for questions, so if you have any live questions, if you're watching this live, feel free to put it into the chat and I will break in and make sure that Amy gets the question. But otherwise, I'm looking forward to sitting back and listening to these reviews and maybe get a little mad because there's probably some proposals that we're not going to like. But I'm going to turn it over to you and let you take it away and give a good summary for us.
Thank you, Lisa. So welcome to the webinar. We are going to discuss the proposed revisions to USP 795 and 797, which came out in September. So I'm not going to be able to give an exhaustive list of all of the changes proposed, but I am going to do my best to highlight the major changes and even some of the minor ones. So starting with 795, let's start from the beginning. As you might have been aware, there were some proposed changes back in 2019 that was remanded in 2020, and a new 795 chapter has been proposed. So basically, as I mentioned, we're going to briefly go over those. And I just want to also just globally point out that a lot of these changes were made to clarify certain things that were on the current chapter, but there's clarifications made, there's maybe additional information. And of course, the bulk of the changes focus on BUD assignments or limits. and this new concept called water activity. Otherwise, the 2021 proposal for 795 is fairly consistent with the 2019 remanded version. So if you had any hand at reading that previously, then you might be in a good spot with this proposal. So anyhow, the first section of the 795 chapter, it's the introduction and scope. So there's a few main changes to look into. I want to take a step back and just, as a non-sterile compounder, if you're wondering what is a non-sterile compound, it might be basic to you, like an oral suspension or a topical cream, but I just wanted to point this out that it does specify otic, nasal irrigations, including, routes for the mouth, rectum, sinus, cavity, sort of local preparations. Those are what are known as non-sterile. And then in a minute, we'll get into sterile, but I just wanted to clarify that so that you guys knew, these types of things are included as a non-sterile preparation. So in the same section, they talk about personnel and settings, and they added something on dispensing requirements. So basically it just says that we need to be proactive to identify and remedy any potential problems, not just when you're preparing and storing and packaging your compound, but even dispensing and transporting CSPs or non-sterile preparations. So we basically just want to follow applicable laws and regulations when we're compounding. And we also need to now designate someone that's going to be what's called the responsible person, or the designated person that's going to oversee non-sterile compounding operations. And I'm not sure if you guys had any interest or background in hazardous drug compounding, but they've taken that whole thing out of this new chapter, and they are referring you to go to USP 800. And for radiopharmaceuticals, that's going to be chapter 825. And there's another thing that they also take out, anything regarding administration, which will be a much more interesting topic when we get to the sterile portion of this discussion. But anyhow, if there's anything else I want to say, basically, like, there's some other definitions. I was in a board of pharmacy meeting one time, and someone was asking, is flavoring a manufactured drug product considered compounding? And absolutely, that is an example of compounding. Splitting tablets is not, but flavoring would be if you just have like a regular manufactured suspension and you want to put it like a cherry flavor into it, that is considered compounding. And essentially it's because these flavors, they're multiple component systems. They're really complex, and they actually do interfere with the stability of a product. So I just wanted to kind of point that out, sort of a sidebar, but I think it's important to understand that. The next section talks about personnel qualification. And there's a couple of things that we want to point out here, in that basically the chapter gets a bit more descriptive in the training and competency requirements here. It clarifies that all the personnel involved in compounding or in the direct oversight of preparing and dispensing must initially be trained and must demonstrate competency before being allowed to perform their job duties. So translate that. That means pharmacists out there, you need to be understanding and actually competent in these operations before it even says here, before you're dispensing. So you want to make sure that even as a pharmacist, you can perform, you know how to use that mixer, you know how to use that analytical balance. You understand a lot of these, principles that we're talking about in compounding. So just essentially know that if you're supervising compounding, you need to be trained and competent before you do your duties. And then you need to repeat these kinds of trainings and competencies annually. So that basically just needs to get translated into your operations and procedures. And I think that's all I need to say on this slide here. So personnel.
And really quick, Amy, I have a question that's come in. So for pharmacists, if you had some sort of training in pharmacy school, you have like a compounding lab or something like that, is that history okay? Or is there something that you have to do more recent before you can go in and start dispensing the compound? So there was a question of that school training versus maybe on the job training.
Well, I have to say, it's nice that your school actually trained you. A lot of schools don't even have these sort of formal trainings But that's good to hear that that's at least happening now than back in the day when I was going to school. Well, okay, so to back it up, you need to be trained and competent in your own facility's procedures, okay? So maybe you learned generally how to make a suspension in your, your school's compounding lab, or even if you went to like a fancy lab in somewhere in America that was whatever. But essentially, if I can answer this right, you need to be trained and competent in what you do at your facility with your equipment, your specific procedures, and your personnel. And so I hope that answers your question. There's nothing wrong in having, and it's actually really good to have sort of a knowledge base going in before you start your role at this facility. But when you're in this new role at this facility, you really want to be competent in what you do there. I hope that answers that.
Yeah, I think that answered it perfectly. You need to be trained on your equipment in your lab, because you might have been trained at an EMP under a certain brand, and
Yes.
now you have a different brand in your lab, that
Absolutely.
kind of thing. And so best to go through all of that training and documentation first before you start making and dispensing products.
And that's one of the things that Two Trees Consulting can provide, is help come in there, administer, maybe if you're doing sterile compounding, administer the media fill, or actually go through your procedures, show you how you're doing your stuff there and do it right. So there might be some sort of like a guided training that might be interesting for practice sites to implement. I don't know if that's something interesting, but okay, moving on. I have a slide up here on the next section called Personnel, Hygiene, and Garbing. So the things to remember here are that there's a section called Personnel Preparation. And this discusses the appropriate hand hygiene and cleanliness. And basically before it was like, thou shalt not have any jewelry on or piercings or what have you. And you don't want to have those things. That's true. You want to, make sure like your nails are trimmed and clean and all of that. But really what they're trying to say here is they're trying to give a provision. So like, let's say like you have a wedding ring and it's like on there, that thing is not coming off. There might be a provision for you that as long as you're not going to disrupt the environment, that you can maintain the environment specifications for bioburden, that you might be able to get this provision. The one thing to keep in mind though is that needs to be documented. So if it's like Amy wears her wedding ring when she goes in to do some non-sterile compounding and it doesn't come off, then I'm going to get this written provision in my training folder or my own personal file, and that'll say that. And there might be some kind of assessment or risk assessment saying the fact that she's wearing this doesn't affect it because it's covered properly with gloves, something like that. So another change that was come in was the garbing and gloving requirements. It adds that garb should be removed when leaving the compounding area. So don't just, keep your gown on and go eat lunch with it on. Nope, don't leave the area with it on. You can hang it up and you can reuse it for the remaining of the shift, but just don't let it leave the area. I hope that's clear. Okay, next, building and facilities. This was, from the current chapter, maybe there's not so much guidance in this, but this 2021 proposal is making sure that that there must be a designated space for non-sterile compounding. The area going to wear a gown. It's really hot. So just to make sure that what to wear and what sort of the do's and don'ts in that facility. So okay, next topic is cleaning and sanitizing. There is a new table and it just discusses the minimum frequencies. It just becomes more defined. It says like, clean and be sanitary before compounding, after each compounding, clean your floors on a certain daily basis. And maybe when there's spills, I just recommend you to go and study it to get those more detailed examples of what they want. But otherwise, it's pretty straightforward. Equipment and components. So there's a few interesting things here. Weighing, measuring, other manipulations of an API, which is that active pharmaceutical ingredient, or like a powder, right? So if you're working with powders, especially ones that can generate airborne contamination, you really need to be compounding in something like a containment ventilated enclosure. So these things, they must be certified annually. And, there might be some questions like, well, do I need to compound in this this hood, if you will, for everything. And USP, they do have a lot of, basically they want you to conduct a risk assessment for answering that. So you need to sort of figure that out and see, should I or shouldn't I? But at least for powders, you need to be putting those in, weighing them in a containment ventilated enclosure. And so new thing that's spelled out, at least from the current chapter, is that APIs must comply with the USP monograph if available, and they must be manufactured in an FDA registered facility, and they must be accompanied by a valid C of A. So don't just go to the grocery store and buy that excipient or, any active ingredient. Excipients, they should be obtained from a registered facility by the FDA, but really the federal and even state boards of pharmacy are cracking down on this. They really are concerned with what materials are you working with. So just be cognizant of that. And then there's another interesting thing: packages without labeled supplier expiration dates on that actual container. You need to put a shall not use beyond 3 years from receipt. So then you also have to know when you received it. So you should put on the label, received on whatever today, let's say October 14th, can't use it beyond, 2024 of this date. So
Amy, quick question on those suppliers. So I know back in my pharmacy when I was doing vet compounding, and I know obviously a lot of people do vet compounding now, they use, for example, packaged peanut butter or pre-packaged dog treats that they then, pulverize and like put the active ingredients into. Can you use food type products like that in order to use in compounding? Or does it have to be from an FDA registered facility if it's okay in the food store and you're going to add and manipulate it to add the active in it?
So as it's written here, if it's the active ingredient, so that's not this case, but if it was your active, it has to be from an FDA registered facility. If it's your excipient, like a peanut butter or something like that, it just says should.
Okay.
So if you are going to get it somewhere, how do you know that quality of that peanut butter? I know now we're getting into is this peanut butter going to turn rancid within 30 days of opening, or how long has it been? I don't know those nuances, but the general and best answer, especially as a consultant, I don't want to blindly say, yes, you can do this, it's okay. I can't tell you that, but I would do what's like a risk assessment and make sure that you have properly qualified the supplier that you get this peanut butter or this non-API ingredient from. And you've looked at their quality system, even from like a survey. And this is a whole other discussion, by the way. I could talk a whole hour about how to qualify a supplier and what to look for maybe that's a good topic for another time.
Yeah, but, absolutely.
This is, and it is a hot topic. I will tell you that the boards of pharmacy and the FDA, they are really concerned, and not just with non-sterile compounding, but sterile compounding. They basically have a document called Know Your Supplier. So it just means like if you're going to use something, especially for APIs, but I would even argue that for your excipients, anything that's going to go into this compound, you've got to be confident in its quality. So that's the sort of global generic answer. And I know that's always hard for someone to hear. They want to know exactly what to do, but I can't give you more than that. We might need to dig in more and help you more specifically to answer that question. But it's a good question. Thank you. So we're on to master formulas. There wasn't much here. It's just basically you need to have a master formulation, and you need to have a compounding record in place. And there's just a box. It's a nice little visual that you can go and check out. Next is release inspections. So this section basically talks about what's required to release your compound. There's not a lot of content changes here in this section, but the things to remember are that you have to confirm that your non-sterile preparation, that the labeling matches your compounding record, right? So that it is actually what you intended to make. And then you need to conduct some visual inspections on these preparations. So determine if the physical appearance is as expected. and then actually perform a visual inspection of that container closure. So like if it's like a suspension, it's not just open and potential to leak or something like that. It doesn't need to be like a USP 1207 container closure integrity test. It's just a visual confirmation that, yeah, this is closed well and it's not going to leak or open, such like that. And then there's other things that you need to assess per each compound. So there's no global answer for you, but like things that you might also be interested in, pH might come to mind if you're doing like a liquid or something like that. Eventually you want to study, does the thing that I've made, is it meeting the potency requirements? So conducting an assay test on that might be important, but you need to, again, conduct a risk assessment to see how often or for which ones you want to do that for. Labeling. I mentioned a minute ago that you need to make sure whatever's on your label needs to match what was actually made. This section just adds clarification that the label on each container, it must display certain information prominently and legibly. And then it also added that if you can't fit everything, then there's something called labeling, which is like the outer package. That whatever you can't fit on the actual immediate containers label, you can use like an outer packaging label to add to that. And then, of course, I mentioned a minute ago about checking the master formula and the compounding record against the medication order against your label, and all of that should be cohesive. So I hope that makes sense. Okay, now the moment we've all been waiting for is establishing BUDs. So this is where the bulk of the 795 changes came about, and a lot of the time was spent here since that appeal happened. And, section 10.1 and 10.2 didn't really change much, but it basically talks about how expiration dates apply to manufactured drugs, and BUDs, or beyond use dates, apply to compounded preparations. It explains that the studies for conventionally manufactured drugs are much more exhaustive than even those for compounded preparations. And then it also broadly states, section 10.2 states that parameters to consider when establishing a BUD are basically things that can impact the stability of your preparation. So they talk about something called water activity or water content. That sounds right in our heads, but like water activity has a definite effect on stability. So does chemical and physical stability, compatibility with the container closure system, degradation of the container closure system, microbial proliferation potential, which will happen more if you have a high water activity compound that you've made, and even deviating from the essential compounding instructions, that can even affect stability. So I just wanted to mention that no freelancing. If your compound says, these are the steps in this order, you need to follow it that way. Otherwise, it might even affect your stability. So here is the table that is probably one of the most looked upon areas of the new USP 795 proposed chapter. This is the one that everyone goes to immediately, and they're wondering, what's changed and what do I need to know? So I would say that, BUD limits are explained by the type of preparation in the absence of a USP compounded preparation monograph or a specific stability study. So in other words, these are your limits. You're not going to go beyond these, especially in the absence of a stability study or in the absence of a USP compounded monograph. And I want to note that these default BUD limits were not really changed from the 2019 remanded version, but the committee basically, they gave a lot of rationale documents as to why these limits are proposed. And they, I guess one such rationale that I want to talk about right now is the concept of water activity. So this term water activity helps us figure out if a compound is considered aqueous or non-aqueous. Because before it was like, oh, for non-aqueous preparations, the BUD is this. Well, what is aqueous? What is non-aqueous? That's what this term is going to help us answer. So if you know which category that your compound falls into, then you can use this table in a meaningful way. And USP does provide a table, and I'll give you an example in a minute here about some examples of what that looks like. So for aqueous dosage forms, they're defined as having a water activity of greater than or equal to 0.6. And this basically just means that it has a lot of free or available water in the compound, the finished compound. And these compounds, they participate in hydrolysis reactions. They support microbial growth. So conceptually speaking, they're going to have a shorter BUD, right? So emulsions, gels, creams, solutions, sprays, or suspensions with a water activity of 0.6 or greater, they're going to fit into those aqueous dosage form categories. And then, so looking at the top half of this table, that's basically what it discusses is the aqueous dosage forms. And then you can see that within the aqueous dosage forms, they categorize them into two types, basically the non-preserved and the preserved. And so they're given a shorter BUD limit than non-aqueous because of, conceptually what we mentioned about microbial growth and being more reactive. And so on the lower half of that table, we're looking at the non-aqueous dosage forms with a water activity of less than 0.6. And so these include, again, it can be a liquid, it can be another non-aqueous formulation like a capsule, a tablet, granules, powders, suppositories, troches, topicals, etc. But the kicker is it's less than 0.6 water activity. So oral non-aqueous liquids, they're assigned a 90 BUD day, and basically for the others, it's a 180-day limit. And liquids, conceptually, they get a shorter limit because of degradation kinetics. They happen a little bit faster than solids and semi-solids. And so this this is basically a risk assessment that the committee settled on. They didn't just, they didn't supposedly dream up these. They really took a lot of thought into here's what we need to settle on and here's why. And so that's hopefully in your mind, you're seeing that there's a little bit of more flexibility for all of the preserved aqueous preparations as compared to the current chapter. And maybe you can see that there's a little bit flexibility for the non-aqueous as compared to the remanded chapter. I don't know if you'll feel that way, but that's I think that was their intention. And so it's important to remember that these are maximum limits. Okay, you don't just, I'm making a non-preserved aqueous dosage form, so I'm going to assign it 14 days. You need to actually consider that there might be another reason to make it shorter. So you just need to keep that in mind and do your due diligence. Your board of pharmacy will certainly expect that of you. At least they do over here in California, where I'm based out of. So water activity, I promised that I would give you a little more info on that. USP gives you a nice little table, and so I basically just sort of summarize some of the things that were on here, but the description for some of the top examples of non-aqueous. So you might see like a lollipop generally has a 0.46. So that is less than 0.6. So it's under the non-aqueous category. And then here's some other examples of aqueous. So maybe you've got like a cream, it's an oil and water emulsion. It's 0.96. So, and that's interesting here. Look at this emollient cream. Even if it's in petrolatum and mineral oil, it's still made it up to 0.98. So just sort of study these. And I know your question then might be, do we have to actually be testing these? And no, you don't need to test them. I think it even says that in the chapter, but I think if you were confused or were actually wondering and really not sure, then you might want to test it. And not might, you probably should test it, especially as a consultant. I don't want to tell you like, you don't need to do that, but you really should consider if you can't back up what you do with science or a reference, you need to do something to show them what is the water activity. How can you assign it if you don't know? So I guess one thing that was coming to my mind is, some of these new proprietary bases that certain suppliers might be touting, I would just start looking into that more and seeing like, is that proprietary base, is it really non-aqueous? Like the finished compound that I make, is it really going to be a non-aqueous thing or is it? I don't know. You might need to conduct an assessment on that. So the next thing to remember is what I was hinting a minute ago, that shorter BUDs may be required, okay? Just because we have these limits to refer to doesn't mean that it's always going to be, stable for up to that time. So essentially, let's say you have a component that might have a shorter BUD or expiration than your compound. You need to assign a BUD that reflects that shortest BUD or expiration of that component. You follow me? And if you know that your preparation is prone to rapid degradation, like let's say in a powder form, it's good and the capsule can go for 180 days, but once you put it in water, maybe it's a rapid degradation and it's known, maybe there's, it's well known and documented in the literature, you should really assess, should I be assigning this BUD limit per USP, or do I also need to consider some of the actual properties of that API. And if you are certain, ingredients are sensitive and you don't have the data, the default limits, they don't protect you from performing your due diligence. So in other words, you're gonna get asked by, your board of pharmacy to explain yourself. How did you arrive at this? There should be a rationale. Even in your master formula, it says that you need to provide the rationale for the BUD, the reference. So how about assigning a BUD from a USP monograph? You basically, there's several USP NF compounded preparation monographs that have been studied for stability, and you can reference those and assign the BUD from there in place of those limits that we just previously discussed. So this means if you're compounding from that USP NF formulation and the directions, then you are assigning the BUD listed in the monograph, and you're not going to exceed that. Now, extending BUDs with stability information. This is pretty interesting here. So what about if you've conducted a stability study or you want to refer to one from a literature? There's a few things to keep in mind. If you study that table, of the actual limits, we're limited in some way. But even if you're going to conduct a stability study, the committee concluded after much deliberation and assessment that your stability study must be stability indicating, and it is required to establish an extended beyond use date. In other words, potency over time testing doesn't cut it because it doesn't differentiate from the compound of interest from the degradants and excipients. So it's definitely expected if you're trying to extend the beyond use date of a non-sterile preparation. But what we see here in this proposal is even if you do conduct a stability study on your compound, you will be limited to this 180-day maximum. So even if you see that it's stable for a whole year, there's a cap that's put on. And this cap was put here for basically a variety of reasons. But the committee stated that, in 503A compounding operations settings, we're supposed to be doing this for a specific patient that's intended for immediate administration or even short-term administration following storage. So really, we're starting to get out of the scope, and we're not following heightened quality standards that GMP facilities follow. And even though the stability studies are, from stability indicating methods, they're still not as rigorous as those conducted in a GMP setting. And so for these reasons, the committee did put a cap on 180 days for extending the BUD. And for aqueous non-sterile compounds, so you're going to need to also perform an antimicrobial effectiveness test. And that needs to be conducted at least at the end of the proposed BUD in order to extend it to validate that your preservative system is still working and you don't end up with a contaminated product. And it's not uncommon for preservatives to be inactivated by a pH change or reactions that can occur with the preparation or container closure system itself. So for aqueous preparations, those that have a water activity that's equal to or above 0.6, they already provide an environment where microbes can proliferate. So if you would be able to do like a bracketing study for your compound if you carry out, let's say you want to have a lot of different, testosterone cream, you carry a lot of different concentrations, then you would study the high and the low, and you would see that the preservative system also works on these extremes. And so then you can, hopefully save some investment funds there. We still have a lot more to cover, so I want to keep moving if possible. USP 797. So we finally made it. Let's talk about these proposed changes. So something very interesting here about, actually, hang on a second. In my notes, I have it. The definition of compounding. I wanted to start by saying that the definition of compounding in USP aligns more with the FDA's 503A definition, meaning it doesn't include the concept of administration. Okay, so as per section 1.2, if you're going to go and look this up, I'm reading it here from my notes. It says, for the purpose of this chapter, administration means the direct application of a sterile medication to a single patient by injecting, infusing, or otherwise providing a sterile medication in its final form. So administration of a medication is out of the scope of this chapter, okay? So I also want to talk about this section 1.4, which states compounding does not include mixing, reconstituting, or other such acts that are performed in accordance with the directions contained in approved labeling provided by the product's manufacturer and other manufacturer directions. So essentially, preparing a conventionally manufactured sterile product in accordance with the directions of that approved labeling is out of the scope of USP 797. And you got to meet a few things. So it has to be a single dose for an individual patient. The approved labeling has to include information on the diluent, resultant strength, container closure system, and the storage time. So let's say you're going to go off, let me just give you an example so you know what I'm really saying here. Let's say Remicade. The labeling says that you're going to reconstitute a 100 mg vial with 10 mils of sterile water for injection. Then you obtain a concentration of 10 mgs per mil. You use a syringe with a certain gauge needle. You dilute it into a 250 mil sterile, normal saline bag. And the infusion should begin within 3 hours of reconstitution. This is what that FDA labeling says. And if you do this, this is not called compounding. This is called administration. And it's out of the scope of 797 now. So infusion must be administered intravenously for, at least it just keeps going on with that example. But I just want to point out that it's not considered compounding anymore. I think people might have some major concerns about quality here. And then others might want to think that this might be easier, like a way to avoid some of these regulations or standards rather. But just be careful here. And, USP is not a board of pharmacy. They are not the FDA. They're not the ones that are regulating you. So just make sure, even if this chapter does become actually approved, is your state board going to also align with this? Because maybe they won't. But that was just something interesting that I found in just the definition of compounding has been changed. So the overview, I put this slide in here just kind of as a general overview, but most of the changes as in 795, they're focusing more on the BUD assignments. There's there's lots of minor changes that are made throughout all of the sections. But really we're all basically just focusing on these BUD changes. So what's outside the scope, similarly as in 795, with 797, we're not going to be talking about hazardous, we're not going to be talking about radiopharmaceuticals, and now we're not going to be talking about administration of compounds, sterile compounds. So, and then other things to note, repackaging, that has a whole other chapter to look into, and that isn't, that's out of the scope of this as well. So immediate use CSP requirements, there's a couple interesting things to remember here that basically compounding CSPs for direct and immediate administration is not subject to the requirements for category 1, 2, or 3. And you know what? I feel like I did a disservice and I didn't explain myself a little bit here. So let me back up here. So essentially, this new chapter, they've got something called Category 3, and I will be introducing it a little bit better soon, hopefully in my notes here. But essentially, it's going to be a lot to do. So, talking about immediate use CSP requirements, basically, we have to follow aseptic technique and processes. They got to follow the SOPs. You got to make sure that you prevent mix-ups. One new criteria that was added in this proposal is that personnel preparing the immediate use CSP, they must be trained and demonstrate competency in aseptic technique and processes as it relates to this, because, even if it is in an emergency situation, you still want to preserve and not be so, what's the better word for dirty, but you don't want to add bioburden. You don't want to make something that could be sterile into a non-sterile thing just because you're in a hurry. You want to be able to follow those appropriate processes that you've been trained and competent in. So the preparation is performed in accordance with evidence-based information for physical and chemical compatibility. So you're following some kind of reference here. The preparation involves not more than 3 different sterile products. Any unused starting component from a single dose container must be discarded after preparation. So a lot of this is still, similar to the current chapter. And then one thing to note here is that administration must begin within 4 hours instead of 1 hour following the start of the preparation. So it seems like there's a little bit of flexibility added here with regard to that. But otherwise, most of this was the same. So now I would like to introduce the concept of Category 1, 2, and 3. So the current version of 797, as you might recall, it categorizes something called low, medium, and high risk CSPs. And then the 2019 proposed version, it introduced this concept of Category 1 and Category 2. And these categories did not change, but they added this new category called Category 3 that's a bit distinguished. And essentially, these categories are primarily based on a risk assessment that accounts for the state of the environmental control under which they're compounded. So basically, how dirty or clean is your environment? The probability of microbial growth during the time that it's going to be stored. So how long is this thing going to be stored? The time period in which they must be used. And so the 2021 proposal is a bit different than the 2019 in that here's this new, Category 3. So essentially, Category 3 CSPs, they get to enjoy extended beyond use dates for up to 180 days, but they need to meet additional requirements. And there's a lot of them. And they're mostly in alignment, honestly. They look a little bit like CGMP requirements that 503Bs have to follow. But for example, if you're going to go into Category 3, which again, if you're trying to extend your BUD, beyond the limits, you need to be testing sterility. You need to test endotoxins if applicable. You need to perform personnel qualification more frequently. We're talking every 3 months. You must use sterile garb, all sterile garb. You can't show any skin in that room. Sporocytes for cleaning must be done weekly. There's a much more intensive environmental monitoring program, and you have to confirm your stability through a stability indicating study. So this is a lot of hoops to jump through. There is a cap, and so basically you can't go beyond what that cap is. The committee didn't want it to be open-ended, kind of much like what we talked about earlier. So here's sort of a breakdown for personnel qualification. And I tried to, show kind of what we're living right now versus what was proposed but got remanded versus what's proposed now going forward. So, comparing the chapter to the 2019 proposal to the 2021, we basically see that we need to perform personnel qualification every 6 months for Category 1 and 2. And Category 3 is every 3 months for personnel who compound those Category 3s. And this gives compounders an opportunity to identify the deficiencies in garbing and compounding process to mitigate potential contamination excursions, and even the CSP sterility failures. So taking a look at the chapter, we see that USP distinguishes the hand hygiene and garbing competency from aseptic manipulation competency. And you got to note that there is a zero tolerance for CFUs recovered from the hand hygiene and garbing competency. So we must show that we can gown up and that we're not going to contaminate our garb, and that for, the aseptic manipulation competency, this includes the media fill, the glove fingertip testing, and surface testing of a work surface at the completion of the media fill. So you can have up to 3 CFUs for both. And again, training and competency applies to pharmacists that are overseeing the compounding operations, not just technicians, the ones that are doing it. So that's something to keep in mind. Let's move on to minimum garbing requirements. So the minimum garbing requirements for preparing Category 1 and Category 2, they include the low lint garment with sleeves that fit snugly around the wrists and an enclosed neck. So those are your gowns. Low lint covers for shoes, low lint cover for, the head. That also covers the ears as well. And if applicable, facial hair cover, low lint face mask, sterile powder-free gloves, and so basically that's pretty much the changes there. And so going on to the minimum requirements for category 3, this is pretty interesting. So if you are doing category 3 CSP, so basically, in other words, you want to make a sterile preparation with a longer beyond use date than those limits that we're going to discuss here in a minute. There's additional garbing requirements, and you have to meet them at all times. That's the kicker, at all times. So this goes for all personnel, regardless of whether CSPs are even compounded on that particular day. So if you're doing category 3, exposed skin in the buffer room is not allowed. Your face and neck must be covered fully. And you take a look at the picture to kind of really get the idea here. So there's no exposed skin allowed. All low lint garb must be sterile. And disposable garbing items, they must not be reused. And if you're doing a laundered garbing service, which might be interesting to implement, they need to be using a validated sterilization cycle that you review and make sure that it's appropriate. So let's check out our facilities. So there's a minimum PEC placement. If we're looking at category 1, it can be located in an unclassified area without an anteroom or a buffer room. And that's when you're going to do those category 1 CSPs with those shorter beyond use dates. And for category 2 or 3, your CSPs, the PEC or your hood must be located in an ISO class 5 positive pressure buffer room with an ISO class 8 positive pressure anteroom. You can make this more strict if you want to have an ISO 7 anteroom, that's fine. But these are the minimum requirements, right? And so in this picture for category 2 or 3, you don't have to do it this way exactly. You can even have an anteroom that's clean and an anteroom that's dirty. So there's some special things that you can do, or you can just have a line of demarcation like it's shown here. And then also there's a couple little PEC or primary engineering control terminologies, just to make sure that you guys understand that an isolator, when we're talking about isolator, it doesn't just mean your typical glove box. This is the kind that like big pharma uses, and they've got their own decontamination systems built in, and there's it's basically a full enclosed system to the outside world. So that's a bit different than what we had previously defined in the last chapter. So microbiological air and surface monitoring, let's talk about that a bit. I did a quick comparison here as to the current versus the remanded versus the newly proposed. So there's no major changes to these procedures for viable air and surface sampling from the 2019 proposed version. For categories 1 and 2, the viable air sampling, they must be conducted in all classified areas every 6 months, and surface sampling is conducted monthly. Okay, so for category 3, this is much more intensive. You need to be doing that viable air monthly, and your surface sampling in each classified area must be at least weekly. Additionally, for category 3, a surface sample must be taken inside the PEC after compounding each batch, so and prior to cleaning and disinfecting, so no cheating. And category 3 is every 3 months for personnel who compound category 3 CSPs. The definition of a batch is also important to understand, and there's a glossary term for that. And additional samples basically tell you what's going on in the PEC from a contamination standpoint. So during the time of compounding, so a lot of the language is very similar, but you really want to make sure that you have passing test results. So that gives you confidence that your PEC was operating under a state of control and that personnel were exhibiting good aseptic technique, if that makes sense, during the time of compounding. So keeping in mind, microbiological identification testing would now only be required if the results of those samples were out of specification. So let's see here, cleaning, disinfecting, and applying a Sporocyte. I want to talk quickly about how basically things must be low linting and they should be disposable anything that's used in your PEC must be sterile. You need to basically designate your mops or your reusable types of tools, and just dedicate them and show where they're used. And then the Sporocyte for category 3, that's been modified in this proposal, so you need to apply that weekly instead of monthly. So it's a bit more intense. Master formula and compounding records. I'm trying to go quickly here. So compounding records have similar requirements, and they are recorded just as the prescription and medication order or label. So all that needs to be cohesive, but it's a nice little box that you can go and check out to see what's required. Let's move along. I really want to get, so visual inspection, just know that you have to visually inspect your CSP and verify that it's of appropriate quality. So, make sure and that your final preparation is good. So sterility testing, some category 2 and all category 3 CSPs require sterility testing according to USP 71. If sterility testing is performed, the minimum quantity of each container to be tested for each media is specified in that chapter 71. And then table 2 and the number of containers required to be tested in relation to the batch size. So you can refer to that. One thing I want to point out is that the maximum batch size for all CSPs requiring sterility testing is now going to be limited to 250 units. So this rationale is based on the low threshold for test samples as part of the testing process. Basically, if you understand contamination, you might not contaminate every single vial or every single unit that you've made. Maybe it only happens in a few. So the probability of catching those contaminated units is pretty low. And then you're already only sampling a certain amount. So it basically was, again, another risk assessment from USP where they came up and realized that we need to cap this at 250 units. And beyond this, then you really need to consider, that you're impinging into more of a manufacturing type of setting. So bacterial endotoxin testing. This talks about the requirement for endotoxin testing. It's basically required for category 2 and category 3 CSPs that are prepared from non-sterile components. There's a few other details there to look at, but I really want to get to this BUD part here before we adjourn. So establishing a BUD. Essentially, when establishing a BUD for a CSP, compounders must consider parameters that may affect stability. And so they include chemical and physical stability properties of the formulation, the compatibility of that container closure system with the finished preparation in there. There's leachables, that's a concept. There's interactions, there's adsorption, there's extractables, there's, all these things can affect stability. So, and with regards to sterility factors that affect stability, you've got to consider things that are, your environment. How is this CSP being prepared? Is it in a clean room suite, which is a much more controlled area than an SCA? The aseptic processing and sterilization method that you're using, are you using terminal sterilization, which has a higher sterility assurance level than if you're doing aseptic processing, which is like your typical use a filter or go sterile to sterile without a filter. A terminal sterilization method is much more, robust than that. The starting components, are you starting with sterile? Are you starting with non-sterile? Whether or not sterility testing is actually performed or not. And then the storage conditions, what's the packaging and the temperature? So these things affect stability from a sterility standpoint. So comparisons, if we're looking, this is sort of a quick recap or reference for the current USP 797. And just moving along, this new concept, or this from 2019 and now carried over here, the category 1, we've got a room temperature of 12 hours and refrigerated at 24 hours or less. So that's sort of the new established limit for those. Now, category 2 is a bit more involved and sort of teased out, just depends on where you fall. So again, we're from our risk assessment, we're trying to consider, are we autoclaving these and it's a terminal sterilization sequence, then that's going to have a longer beyond use date. If it's, from filtration or it's just sterile to sterile, that's going to have a shorter beyond use date. So study these, they are the limits. They are not just your defaults to assign. And, it's just important to understand the concept of how these came about. Category 3 is the major change here from the remanded chapter. So this didn't exist in 2019, but here we are, 2021. This is the proposed revision, the major change here. So if you are going to be doing aseptic processing of a category 3, you have to conduct those sterility tests. Your cap at room temperature is 60, refrigerated is 90, frozen is 120. And if you are autoclaving or doing some other terminal sterilization technique, you can go up to 180 days. But remember, there's all these new hoops to jump through. So these additional requirements, they include, you have to test the sterility, you got to test endotoxins if applicable, frequent personnel qualification, you got to have that sterile garb, you got to be covered head to toe, so that's the bunny suits, and you have more frequent sporocidal use of weekly and a much more frequent EM program, and that stability indicating method's got to be on board. And remember, don't go beyond 250 final units. One of the last slides here, I promise I'm almost done, is multiple dose CSPs. So a couple things to remember here. It just talks about aqueous multiple dose CSPs. You need to do this thing called antimicrobial effectiveness testing. So that's your preservative. You're testing that your preservative system actually works. But anyway, the CSP, it must be used within the beyond use date limit and or up to 28 days after the initial puncture if supported by your USP 51, which is the antimicrobial effectiveness test, whichever is shorter. And then if you're doing the ophthalmics that don't have a preservative in it, you must be within the beyond use date limit. And then, beyond when you open it, you got to use it within 24 hours if stored at room temperature, or 72 hours, whichever is shorter. So submitting comments, I know I'm racing through to get done. USP is, I feel like they're doing a good job in trying to get the word out that comments are happening right now. So everything that I just mentioned, if you don't like something, if you're confused by something, you want more information about something, or you just have a better idea about something, submit your comment. You have till January 31st. I gave a couple of URL links here, but you are welcome to go and submit your comments through that portal. And they'll ask you your information, and you need to be very specific in your comments so they can actually take action or consider it. Now, remember, these changes are not official. They are not enforceable. We are still in this comment period. I would expect that USP, as usual, has to read all of the comments after they are provided. That could take a few months. I don't know how long that would take, but then there's going to be another period. So I guess if I were to, bet, I would say maybe by late 2022 would be the earliest that these would be implemented. But there could be reasons for not. We need to see. So I guess at this time, I know some questions were asked during, but if there's any other questions, I'd be happy to try to answer. I know we're pretty much out of time, but please go ahead and contact us at least at our URL website, twotreesconsulting.com. If I wasn't able to get to your question, or you were a bit shy to ask it in the group, that's totally fine. Just reach out and we will try to help set you up.
Awesome. Thank you so much, Amy. That was a wealth of information. The 7-9 stuff is not my area of expertise. And so I do not envy any of those pharmacies that are doing that. It is a good business opportunity because a lot of people are getting out with, the stricter it gets. And if you're willing to accommodate those, a lot of business can come your way. So, non-sterile is generally where I live most of the time. And you're right. A lot of those were in line with the previous recommendations, but there were some changes, some different changes in there. I noticed, I know the fixed oil suspensions are ones that we used to do a lot of, and those were used to be good for 180 days, but it looks like now under the new guidelines, even though they're non-aqueous, but because they're liquid, they'll be 90 days now. So there's definitely some little nuances, changes in there. And I think the important thing is we'll post the comment link in the actual link in the recording of this, so people can easily find how to make a comment. And please, if you have questions, reach out to Two Trees Consulting. We'll post their information in there as well, so that we can make sure that we're taking care of all of our compounding pharmacies, and that you understand these and you have a chance to comment. Because again, like you said, these are not enforceable. These are not final and approved yet. However, if you don't comment now, don't complain later. You missed your window. So thank you so much, Amy. Thank you, May. Thank you, everybody over at the team at Two Trees Consulting. Really appreciate this. And that website, one more time, is the number 2. So the number 2, treesconsulting.com. And they'll route the questions over to Amy, and we'll get you taken care of. And if you have any other needs for compounding, go ahead and give them a call. They have a wonderful team over there. I definitely use them as one of my go-to experts when I don't know the answer myself. Definitely will go to them. So thank you, Amy. Thank you for doing all this work of putting this together. And I really appreciate your time and effort. And we'll be talking again soon.